She will talk to us about high-resolution correlative 3D imaging of intracellular world under physiological conditions using the synergies of laser light and X-ray microscopes at cryogenic temperatures. And I'm very excited to listen to you, also for introducing us to Cryosim and other nice features. With that, the floor is yours, and I'm happy to listen to you.
Yeah, thank you so much. Can you hear me well?
Yep, that works.
Oh. So today I'll be giving a talk on how we harness the power of two microscopes to image the organelle, image organelle organization and architecture in 3D and 2D. So first of all, I'm going to start by explaining the two microscopes we have. Also, the advantages each one of these platforms hold, a brief overview of our workflow at Beamline B24 diamond lighters, and also the applications of these microscopes in isolation when studying or visualizing organelles, their structure-function relationship, as well as co-localizations of organelles in 3D and in 2D. So first of all, we have two microscopes at the beamline, but there are other ancillary microscopes that help us to do our work. But the two major ones are Cryosin, meaning cryostructured illumination microscopy, and cryosoft X-ray tomography, CryoSXT. So with Cryosin, we're able to get fluorescence information from tagged or labeled organelles within the cellular landscape. But with cryosoft X-ray tomography, we image these cells in their, as well in their near-native state at the water window, where we're able to get natural contrast based on the differential absorption of X-rays by different carbon-rich contents of the cell. And I'll explain this later. So to put this in perspective, we image from tissues, cells, and organelles, and we occupy this resolution scale between 25 nanometers to 250 nanometers, and then we fulfill this resolution gap for our workflow at the beamline. So with Cryosin, like I mentioned before, we get fluorescence information from these cells, where we're able to mine chemical information and localization of proteins or other features of interest. So cryopreservation remains the gold standard of fixation of tissues or cells, in that, first of all, it retains the ultrastructure of these cells, keeps them in their near-native state and happy. By cryopreservation, we also protect the cells or organelles from radiation damage, either from the laser or from the X-ray, and at the same time, achieving immobilization and preventing drifting while we are imaging our samples. Interestingly, we also are able to double the resolution that we would normally obtain from conventional imaging. As well, we can do multi-channel imaging of up to 4 colors to be able to determine localization or distribution or redistribution of proteins. With CryoSXT, again, still cryogenically preserved samples. So we can do Cryosim, like I described, alone, or in addition, pairing it with CryoSXT. And the good thing is that both of them, the cells remain in their cryogenic state. So we maintain our workflow keeps these samples in their cryogenic state. And we're able to image up to 10 microns thick without the need for sectioning and without losing meaningful resolution. Like I explained before, with CryoSXT, we don't necessarily need to tag cellular features with any fluorophores. So these cellular features can report, show contrast just based on their carbon content. Differential carbon content leads to differential contrast, and we're able to determine which of these cellular features you're looking at. So a brief overview of our workflow with CryoSXT. We start with 2D X-ray mosaics, and then we can go to a certain area of interest, either synapses or viruses or organelles. And for 25 nanometer zone plates, the field of view is 10 micron by 10 micron. And for the 40 nanometer zone plates, the field of view is 16 micron by 16 micron. We collect 2D projections, angular projections from -65 to +65, and at different step increase with different exposures depending on the sample and how well they can tolerate it. After collecting this tilt series, we reconstruct them into 3D tomographic volumes, And from there, you can see that we are nicely annotating different cellular features of interest from the nuclear region to the cytoplasmic region. Here we see the mitochondria, some lysosomes, as well as the cell membrane, giving us beautiful contrast based on the carbon content within them. And thereafter, we can decide to segment these cellular features. So again, just to briefly put in context how we move or work at the beamline. So first of all, we have to have our samples. So where are they coming from the source? If they are not produced on site, they have to be shipped to us, to the facility. And then when they arrive, or after we've done our own wet lab here, these samples are deposited on our grids. We use TEM grids as our platform. And following deposition, we either label with fluorophores if we want to do fluorescence imaging with Cryosin, and then we put positional markers, which are fiducials, that will help us to, positional markers help us to reconstruct tilt series into 3D tomographic volumes, or they can help us to correlate fluorescence images and soft X-ray images. After sample preparation, we then go ahead to vitrify these samples, either by plumb freezing or high pressure freezing. Thereafter, we can map them to assess the ice thickness and fluorophore distribution if we are doing SIM. This is followed by data collection at either the Cryosin or the SXT platform. And after, we can reconstruct our raw SIM data. If there's chromatic shift based on different wavelengths used, we can correct it using Cro-Magnon, reconstruct our data, and then post-processing involves segmentation, correlation, or making of montages from maybe different areas collected on X-ray. And also, we can get quantitative data from our fluorescence and X-ray applications. I'm going to give two standalone studies where we've used either laser light or X-ray to study organelles, and then later I'm going to look at how we study them in Synergy. First of all, we were able to look at F-actin organization beyond the diffraction limits. Here, with conventional microscopy, you can see the level of information or detail we get from F-actin organization. However, when we use SIM, we get beautiful super resolved image data sets, and in 3D rendering, you can see how F-actin is occupying space or distributed in 3D within the cell. Again, we're able to use cryo-soft X-ray tomography to study mitochondrial health in control or healthy mammalian cells and infected cells. Here, we looked at how SARS-CoV-2, the viral agent that causes COVID-19, how it affects mitochondrial health and leads to remodeling. In healthy cells, we see these nice thin mitochondria in these mammalian cells. However, following infection, these mitochondria become stout, truncated, and branched. Again, showing the power of our microscopy platform as a standalone tool and how it can be able to tease out organelle remodeling in health or disease states. Pairing these two microscopes, we've been able to use it to study real virus intracellular localization in multivesicular bodies. So here in red, we have multivesicular bodies, and here in green, we have viruses. And with the soft X-ray tomography, we can see how different cellular features coexist on their own without, how they coexist even following viral infection. Merging these two images and then together with this, we appreciate the power of SIM and X-ray, where we see beautiful co-localization of viruses within multivesicular bodies, and also the interaction of other organelles within the cellular landscape. So SIM, with SIM, you see the specifically labeled organelles of interest, but pairing it with SXT, you're able to see other organelles and how they interact with those labeled organelles as well. Another beautiful study we've done is where we've seen the localization of primary cilia in mice chondrocytes. So cilia normally would not give you enough contrast because of its apical location on localization on the cell. So it's located on top of the cell, and again folding on top of the cell, which has a lot of contrast, gives high background, and you're not able to beautifully localize it. However, in this case, we were kind of like lucky because this one is quite obvious here. But with fluorescence images, you can be able to tag these cilia, see how they are localized with centrioles, and which are at right angles to each other. And with fiducials or mitochondria, we can correlate these two data sets and be able to disambiguate where the cilia is located as well as the centrioles right here. Finally, We're able to also look at how another project that we've done is how F-actin interacts with multivesicular bodies. Again, in soft X-rays, you can't immediately or visibly make out which one is F-actin and how the network branches or anything. But when you bring up these SIM data on top of the X-ray data, you're able to localize F-actin. And here we saw, which corroborated other studies, that F-actin localizes or interacts with multivesicular bodies, hence highlighting its role in structurally maintaining the integrity of the, as well as transport and communication. So far I've been able to explain the two imaging platforms we have at Beamline B24 diamond light source, the imaging and the advantages each of these platforms possess in isolation or in synergy, as well as talking about different applications of these projects in different of these platforms in different projects. I'd like to acknowledge my colleagues at Diamond Light Source, Micron Oxford, University of Oxford and France, and I also welcome your questions and feedback. Thanks for listening.